So the examples of the simple lipids, like I said, fats, and then the oils. We have waxes and many other types, by the way. This is not to say that these are the only fatty acids that you have. Okay, then we have the second type, what you call the heterolipids. These ones, like I said, hetero means different. They are not the same. They have other things added to them. And these other things are the ones we are calling the additional groups. It can be phosphorus, it can be amino, it can be another group that is added to these lipids. So that's why we call them compound. So if you can see the name here, phospholipids are examples of the compound lipids. It means we have phosphate group added to the lipid. So we have a phosphate, phospholipids. And these are very important in the structure of cell membrane. If you look at the cell membrane, you definitely see that you have phospholipids there. There are other elements that you also find on the cell membrane, the glycolipids, okay? And then even lipoproteins, they're also there on the cell membrane. And these all are examples of compound lipids, including the lipids, which are very important in the brain and contain amine alcohol. Very, very important in nervous transmission. Okay, this is still to illustrate to you. You may need to check out the structure of the cell membrane and see the orientation of the phospholipids. Here, we are basically illustrating the phosphoheads, and then the phospholipid heads, and the phospholipid tails. Like we said, the head is polar and is water-loving, and we say it is hydrophilic. And then the tails are water-hating, and we say they are hydrophobic, and they inhale the water. Okay, so usually the heads are facing the aqueous environment, and then the tails are avoiding the aqueous environment. And that's what we have here. These are the hydrophobic tails there. Derived lipids. These are lipids that are derived from simple or compound lipids by hydrolysis. Okay, when you break into smaller components, that process is hydrolysis. So sterols, these are examples of lipids, derived lipids. The terpenes and then the carotenoids are also examples of derived lipids. The steroids, they are non-saponifiable. This is to mean that they cannot undergo through saponification. If you remember how soap is made, the process called saponification. Okay. So we have a lot of hormones that are actually steroid in nature. If you say steroid hormones, we have the estrogen, and then we have testosterone and many other steroid hormones. The terpenes are actually found in plants, and they are used in aromatherapy. Carotenoids are basically pigments in plants, and we have several types of them here. The xanthophylls, carotenes, and then lycopines are examples there. Lipids generally can be liquid, especially those that you derive from plant sources, but they can also be non-crystalline solids, especially those that we derive from animal sources. Solubility may vary, but largely non-soluble in water, but soluble in solvent solutions. Some of them can undergo what we call saponification. From that reaction of these fatty acids with, if you have fatty acids reacting with an alkali, sodium hydroxide, where you form a soap, okay, and then glycerol. That's the process of saponification. Okay, so they have that reaction. Okay, so saponification actually hydrolyzes the fatty, hydrolyzes the lipid or the fats that you have. Anytime then you repeat saponification. So the other property is what we call saponification, which is the process of formation of salt. The process of formation of salt involves reacting either oil or even fats with a strong alkali. And in this case, you can use potassium hydroxide or sodium hydroxide. That reaction gives you what we call a salt. It can be sodium stearate or potassium stearate plus glycerol. So basically, this reaction splits the oils or the fats into that, you get that salt and then the glycerol. Okay, they are poor conductors of heat and then also the electricity. That's why they are very important in insulation. So how can we test for lipids? We can test for lipids using sodium-3 solution. When you add sodium-3 solution to the sample, you will see a color change. You will have an orange-red color which will appear, and that will indicate presence of lipids, especially the triglyceride and cholesterol. And then the other test that you can perform is the grease spot test. With this test, you you place a small amount of the sample on a brown paper bag, and then you allow the spot actually to dry. For example, the translucent grease spot, when it appears, it will indicate the presence of lipids, especially the triglyceride. And then the emulsification test. With this test, you mix the sample with water and shake, and you observe the appearance of the solution. If you see a stable milky emulsion being formed, it will indicate that you have lipids, particularly phospholipids and cholesterol. So lipids can play a number of different roles. Some of them we have mentioned. Energy gives us a lot of energy. And when you break down lipids, you actually get water as one of the byproducts, and that is what we call metabolic water, which can be used for a number of functions. And then a lot of fat allows organisms that are fatty actually float on water. That is a concept to do with buoyancy. And then you have lipids being a precursor for many important biomolecules. Okay, so for example, vitamin D and even the steroid hormones are made from lipids. Nervous transmission, we already talked about the myelinated fibers. Okay, very important in insulation, but also in transmission of the impulse very, very fast. And then you have some of the lipid components being very important as parts of certain enzymes. And then we already noted that phospholipids, glycolipids, and lipoproteins are actually components of the cell membrane. And then lipids are very important for insulation. And then also we have lipids around body organs, and this is basically for protection purposes, and it's providing that cushion. Okay, so that marks the end of this presentation, where we have looked at lipids. We say they are very diverse and therefore have different functions. The key thing is that they have water-getting tails and water-loving tails. They are formed when you have three fatty, you have glycerol combining with the three fatty acids formed.